mentioned like it's hard to find.
Quick shared resource for Wilmot Cancer Institute. You're in the right place. So I'm John Ashton. I'm the director of the shared resource. Those of you that have done any genomics at U of R probably know. Come on in, but wherever you want to go. You can sit in the front seat. There's plenty of space. Anyone that's done genomics at U of R probably has at least interacted with me or heard my name. But I've been here since 2013 in the Genomics Center. And we're going to talk to you today about how we leverage the Genomics Research Center at U of R to help Wilmot Genomics and what that means for anyone that is a Wilmot researcher, Wilmot graduate student, Wilmot investigator. So importantly, for those of you that don't know, we have Wilmot specific shared resources. The Genomics shared resource, which you're here to talk about today, is one of 7 shared resources. These are the shared resources in totality for those of you that have been coming to the Lunch and Learn that Tracy has been hosting. You've heard about some of these shared resources already. So we're typically talking about genomics, which is down here. Like I said, my name is John Ashton. I'm the director. I've been doing this since 2013. The lab is really led by 2 individuals, Elizabeth Pritchett and Jeffrey Malik, who are both serve as the operations director and technical director. So these are the people that run the lab day to day. You'll interact with them when you try and talk about consultations or research projects. Where's the status of my project? How can I use services towards the goals? So if you don't know these folks already, you'll become familiar with them if you want to do anything in genomics with us. But they really are the lifeblood of leadership of the lab and just putting everyone's expertise on the side. So really, what do we do? So when we talk about genomics, we have a connotation that genomics is really just one thing, sequencing genomes. We don't refer to that here. We think of genomics as a very broad swath of things, from basic RNA sequencing, whole genome sequencing, all the way to spatial omics, single cell sequencing, and then beyond. So here, if you go to our website, you can see here are our major buckets of services. We go all the way from helping you extract nucleic acids from tissues, from cells, QCing samples, looking at RNA quality and DNA quality, all the way down to helping with data analysis. So importantly, the Wilmot Genomics shared resource partners with the Biostatistics Bioinformatics shared resource, which for those of you that came a couple of sessions ago, you've heard about that. It's a really important partnership. It allows us to get the expertise from Derek, who's sitting here, and Matt. So he registered, but he's not here. They really help provide the expertise and the advanced knowledge. So if you come to us and you want bioinformatics, data analysis help, we generally will involve them or directly send you over to them. After the seminar, we'll send you a link as well and the post survey to the actual webpage for the Biostatistics Bioinformatics shared resource. There's an actual web form on there that you can ask for consultations or questions, so you have that contact as well. If you contact us directly, we most likely will just send you to that webpage. But you can see we do a broad range of things. Importantly, there are things that we do a lot of and things that we do a little of. High throughput sequencing is the most that we do, right? And so it's not surprising, any types of genomics or omic analyses that you do these days have some level of high throughput sequencing next generation sequencing. So we do a lot of that. We do a lot of single cell omics, single cell RNA-seq, single cell epigenomics, single cell T cell repertoire, B cell repertoire, all those types of things. So we do a fair bit of that. Spatial transcriptomics, we've been doing for a few years. We're doing more of that. This is really the next level of genomics, omics analyses. And we're supporting that with the various technologies and methodologies we'll talk about in a couple minutes. Importantly, none of any of this can be done without the people. And so this is a lab staff picture from last year. We really are all about people. We want to make sure that the people in our shared resource and across shared resources are experts in their field, conscientious, have integrity, really work hard, want them to be almost an extension of your lab. We want them to want to help you, want your stuff to succeed and things like that. And so I'm showing you here, we have about 20 staff, a mix between lab staff that generate all the data, help with QC, help with the actual data generation. and bioinformatics staff. Again, some of them are directly linked to the Wilmot bioinformatics biostatistics, and some are not, because we're an institutional U of R shared resource. We kind of are on both sides of the fence. But what it says is we have a significant mass of people that all have unique expertise and skill sets. What this means for you is that most likely, if you have a question, we're going to be able to find the answer. Whether it's within this team or the network of people we have out there. Importantly for Wilmot, there are specific needs we want to be responsive to the community. Every year, we have a Wilmot membership survey that goes out. Some of you that get that survey, you know what that is. It's really meant to understand how well are we doing, where are our gaps, or can we improve? So importantly, every year we try to respond to that. This year was actually a very, very impactful year, and we were able to respond significantly to a lot of feedback. One of the common feedbacks we always get is we need to reduce costs. Things are too expensive. We want things to be faster. We want things to be cheaper. So in response to that, we were able to secure Illumina NovaSeq X sequencer. It's the state of the art, the newest sequencer from Illumina. It allows you to sequence whole genomes in 36 hours. It allows you to have the cheapest per base of any sequencing technology. So what this means is we now can significantly reduce our costs for all sequencing related projects and allow us to now support whole genome sequencing, epigenetics, epigenomics, unbiased profiling, things that we were cost prohibitive before because they were just too expensive. Now we can do this with fairly routine, ease, cost effective. For perspective, human whole genome right now with this platform is a couple $100. Last year, it was 1,000. 3 years ago, it was 5,000, right? So it's not linear, but it gives you perspective that now, if you're interested in doing cohort analysis or doing genome-wide association studies or doing things on a bigger scale, sample size is important. You now can achieve a sample size without breaking the bank, right? And that's really important. And what's most importantly is we are the only institution that has this in the region. The next closest is Pennsylvania. Roswell Park does not have one. Very few people in New York City have one. Because of the infrastructure required that we've been working for the last 8 years to build will allow us to, I would say, as you can imagine, a lot of data exchange. And you'll see in a little while our external partnerships. So while we support Wilmot, we support U of R, a lot of our user base and a lot of our collaborators are spread across the country, across the world, which allows us to leverage their demand, keep things cheaper, and to get something like this. So this is something that we've been using. It's in operation now. Most projects that anyone that's starting between now and going forward, will leverage this platform unless it's a legacy project. So if you have a project that you've been doing for 5 years, you'll want to make sure that the platform doesn't impart any cynical bias. So we usually will have those stuff. The other important thing is epigenetics is a very large space. It's something that we have been historically not as supportive of, largely because these are very critical mass and it's an emergent field as well. I'm showing you the results of this current year survey where the majority of people have been saying they want DNA methylation analysis. So like I just said, the NovaSeq X allows us to do unbiased genome and epigenomic profiling. That's great. I won't lie, the data analysis is not insignificant, right? To take that data, while it may be more cost effective to do, it takes a lot of data analysis time and people that actually know and have done differential methylation analysis or motif analysis. Things are very specialized. So we acknowledge that. So in addition to that, we also secured an array-based system that is a targeted profiling system and only captures about 950,000 CpG islands, but it allows you to do very broad profiling of a large swath of regions. A lot of people in public health or population health use this array because they don't care about base pair methylation, they care about big changes because big changes mean something. So we've secured this, we have this, it's coming online in July. What this allows you to do is the data analysis on this is very, very easy. They're arrays have been around for 30 years. Analysis methodologies really haven't changed, but you can very quickly, in a fairly cost-effective way, analyze GWAS and epigenetic status in a large group of samples. Some folks in our cancer prevention and control program have been using this with partners at Roswell and other places. They do this a lot, and now they're working with us, and we're able to support that. So if you have any need for that, again, the caveat is it's targeted. It doesn't give you per base methylation. It doesn't give you anything outside of those CPG islands, but you might not need that. So this is something that's coming online. Again, it's comparably priced now if you want to do an array versus unbiased genome profiling methylation. It's about the same price. The difference is what you get out of it. You'll get way more data from sequencing unbiasedly, but you have a lot more of an analysis burden with that as well. In addition to that, I mentioned that we've been involved in spatial transcriptomics, spatial omics for a while now. It's an emergent field. There's new technologies, new methodologies constantly coming out. In the field of cancer, you can all probably agree, understanding the context of where a tumor cell is relative to the microenvironment, or where an immune cell is relative to a tumor is very important, right? So being able to take a bulk tissue, isolate nucleic acid, and ask questions has marginal value. Being able to take that same tissue, dissociate into single cells, and then ask what single cell repertoire is there has value, but not as much as understanding where those cells are in relation to one another. With new technology, I'm showing here, it's a technology from 10x Genomics called the Xenium. We now can offer single cell resolution spatial transcriptomics. So what this means is you can look at single cell resolution, what expression of transcripts are in a cell relative to another cell. You can ask questions around, I have a tumor, how heterogeneous is that tumor based on transcription? If I have an animal model and I'm trying a new therapeutic challenge or a new antibody target for some specific purpose, how is that tumor responding in totality? Does the microenvironment play a role? This is something that we will be launching in July. It allows you to profile, right now, 1,600 targeted transcripts. And shortly, 10x will be launching a 4,000, 6,000 gene panel. So the positive is single cell resolution, which is great. You have great images, you're able to identify regions. The negative right now is it's biased, right? It's not unbiased, every single transcript you're going to be able to detect. We have other methods for that, which I'll go into in a minute. But the power of this is it gives you the ability to ask context, which is very important. The other thing, if you are interested in unbiased analyses, we've been doing Visium, which is another 10x genomics technology, for a couple of years now. Visium really is, it's basically a slide. You lay your tissue on a slide that has a grid of spots, and those spots have oligos that capture RNA, and then you just sequence it and say what transcripts are detected. That's great, but it's not single cell resolution. You really need to have 10 to 100 cells in the region to get enough signal. Recently, they launched a new version of this, which is HD for high definition. So you can imagine what that means. It's supposed to be single cell resolution. So this is just showing you the difference between the resolution. On the left is the older Visium technology, which is not single cell. And you can see the number of grids, about 5,000 spots, compared to the new one that's many more spots. So the ability here is you don't have to ask any question or know anything about what targets you want to look at. You want to unbiasedly profile a tissue at close to single cell resolution. The negative is this is the image you get. You get an HD image, which is great. You can see morphology. You can overlay expression data, but you don't get the ability to focus in on these images and structures and this nice confocal-like image. For certain tissues, it's important. For certain tissues, it's not. But again, this is something that we'll launch in July. We acquired a new machine. Of course, all companies like to give us machines we have to pay for new technology. And once that machine comes online, we'll be able to support this as well. So really, at the end of the day, we have the ability to look unbiasedly at large swaths of tissue, single cell resolution, whether it's targeted with this type of analysis where you can see cells, or unbiasedly, you want to be single cell resolution, you want to just say what's better. These are also new technologies. Again, for cancer, this is very important. For most of you that are doing cancer research, you probably will have a use for this at some point. Because again, it's not about the cell necessarily or the cell intrinsic, it's about the cell intrinsic and the cell extrinsic. And that's really, really important, especially for immunotherapy. So that's great. We have all these tools, we have all these different layers of data generation. How can anyone help with data analysis? So I alluded earlier that we partner with the Wilmot Bioinformatics Biostatistics, and we have a mix of non-Wilmot bioinformaticians as well. I'm not going to go through this in too much detail, other than to say this is our current mix of bioinformaticians that are affiliated with Wilmot Genomics. This is their expertise. It's not all encompassing, but it's a broad range of things. But most importantly, these are the centers or divisions or departments that all buy into this model and that these 10 individuals support. There's now more than just this list, but the important part here is this team is integral to helping a broad community. And what this means is not only are they helping that broad community, but they're getting different context and different types of draws in their time and different experiences that they bring back to the team, right? It's really a think tank of bioinformatics, which is where I went to the earlier point. There will most likely be an answer that they can find. Whether it's something they've already been experienced or exposed to, or from their travels and who they support, there is some experience that they can pull from that. Important to that, they also are our training education arm. So we acknowledge that not everybody wants to just rely on a bioinformatics person to help their work. They might want to learn how to do some bioinformatics as well. So this team, with support from Elizabeth and Jeff and other folks in the lab, host seminars, workshops, and they participate in graduate curriculum. So what this means is, again, are 10 people enough for this? No. But we often will have seminars every 3 months or every 6 months, specifically targeted to, let's show you how to do RNA-seq, or let's show you how to do epigenetics, and try to start moving the needle so you get some exposure. The graduate curriculum, again, Lauren, who is not here, she is here, does a lot with the graduate curriculum, IND 412, and things that some of you may have taken to help graduate students start to learn how to do some basic things and potentially some more advanced. But the important thing is these folks have a lot of experience. And I encourage you that if you do have a project or an idea or a challenge, to reach out to them. They're very nice and they're very helpful. But they do support a lot across campus. So keep that in mind as well, please. So I've shown you some new services, what we do, who we are. I think it's important to also relate what is our impact. And so this is really just to kind of give you a sense for how we've been growing. So these are demand metrics from 2018 to present, projects and samples. And the real takeaway here is we've been growing every year. We get more projects, we get more samples, both internally and externally. We leverage this to bring on new technologies, to bring on new instrumentation, to make things more cost effective. And importantly, last year, we had 129 individuals from U of R that we supported. And since 2015, we've supported almost 450 patients. Just to give you a sense where we're fairly productive, we're fairly active. And anything that's genomics related, most likely that came from U of R used us in some way. Importantly, we have a far reach. So this is just a microcosm of the institutions that we work with, both in the US and some in Europe. And this is just a list of some of the recent institutions. Princeton is a new one that's not on there right now. But they reach out to us to become the preferred vendor. PIs want to collaborate with us. And this is how we leverage that novacy. Internal demand on U of R would not be sufficient to have that machine here. But now we can lower the price by getting that machine because we have a lot of external collaborators that work with us. And they send us material and we're able to then pool lanes and flow cells to reduce the cost of it. And importantly, this is just a snapshot of some of the impact. A lot of these journals are high impact, but not all cancer. But it's really showing that we're not only just doing steady state standard things, we're participating in immune research, we're participating in COVID response, we're participating in developmental biology. Again, not only in data generation, but also analysis. So it gives you that we have a broad perspective of things. Other things that might be important to you, I mentioned the bioinformatics folks. We have a webpage. We have a lot of information here that's helpful, both what the surface analysis is. So anything that comes through the genomics here, we usually have some sort of primary analysis. And we think that's important because we want you to be able to look at that really quickly and say, is my hypothesis validated or not? Did my experiment work? Is there an issue? So for RNA-seq, for example, it's already through pathway enrichment. So you can get that data, it's free of cost. Usually takes a few more days, which is why it might take some time. But you get that. You get heat maps, you get tables, you get pathway information. But also gives you more information on what if you need more than that? What if you need something outside of that standard analysis? We support that. We call that collaborative analysis, and there's information on there about that as well. We also list our education and training opportunities. So whenever we have a seminar or workshop, we post that there. We're trying to post the slides as well. But importantly, we've also acknowledged that not everybody can come to those workshops. We can't help everybody go to those workshops or seminars or even in the curriculum. Some people need access to something to help them do their own research in their own time at their own pace. So we worked with Wilmot Informatics to launch a research data portal called HyperGen. It's fairly new and only has a couple workflows, but it's meant to be for anybody that works with us or has some public data that they can upload to it and say, I want to run the exact same RNA-seq analysis that Genomics does. You can do that. You can do that very easy. You can do that in your own time. You can do that whenever you're ready. You get the same figures, the same type of output that we would do for you if you ask one of the bioinformaticians to do that for you. The difference is you can do it whenever you want. You log in with your Active Directory, the same login that you use. You can upload, there's no limit, there's no cost. I encourage you, if you're interested, to go here and learn more about it. We're planning to add more workflows to that rate as we go on. Right now, it's whole genome and RNA-seq. But we're adding more components for single cell data exploration and visualization as well, and some other pipelines. For those of you that will be writing grants, which is most of you, we also have facilities and resources text here that you can use for the grants. But importantly, we also have data management plan drafts. So this is something that's not required. And I've been witness to program officers will reject grants if they don't have these now. So MIH actually does care, which is a little bit change the pace from MIH. We have drafts. So these drafts are meant that if you propose to use us for RNA sequencing, we're going to fill out things that we know that will help you. If you use somebody else, doesn't mean you can't use these DMP plans. You just have to change what they give you and what some of the processing metrics that they're on. But these are freely downloadable. We have a bunch of them. We add them as we go. You can use them however you wish. All we ask is if you're unsure about something, ask us. And if you do this, tell people, right? We want to make sure people understand this is here and it's helpful. And this is the website. Again, you can just search Genomics Research Center on Google and we'll be the first hit. But we also, Elizabeth has worked a lot with frequently asked questions. So if you have a question, odds are someone else has, you can go here and start to see if we've answered that for you. And as we get more questions, we try to add it here. So it's a good resource. Lastly, and I'll leave it open to questions or discussions. Where is our value in genomics here at U of R at Wilmot? So we participate in the entire life cycle of the project, not just experimental consideration or design, not just data generation, not just data analysis. So we view ourselves as really the circular entity that no matter where you want to plug in, we're here to help. We again strive to make this be our project too, to help you so that we have at least some desire to really help you push it across the finish line. And most importantly, we have these core values. And so it's really important when you think about what core values would you want from a shared resource, right? Not just genomics, but in general. You want them to be professional. You want them to have integrity, leadership. You want them to be leaders in their field. You want them to be loyal to you, right? You don't want somebody else that's loyal to other investigators or other entities. You want accountability. If a mistake happens, we're going to own it and try to fix it. And you want reliability so that mistakes don't happen all the time, right? So these are what we feel are our core values. And those core values feed into every aspect of the life cycle. And importantly, this is just one of the questions on the registration survey was, how do we engage you? What do you do? And things like that. So I put this in at the last minute to give you a sense of, if you want to use us for the entire life cycle, it starts with the initial meeting. And then we could do the extractions, assess the quality for you, give you your perspective, do an RNA-seq or whatever library preparation, assess those libraries, do the sequencing, and do the analysis. And if we're good at our job, we meet here, and we come back at the end with all the QC and say, here's the project, here's the analysis, or there's an issue, right? If you don't hear from us, everything is working great. If there is an issue, you should expect to hear from us and say, here's the issue, here's the problem, here's how we can fix it. What do you want to do? Again, sequencing can take on many modalities. We have a multitude of sequencers, but most importantly, quality assessment is the biggest thing that we focus on because we have integrity. We want to be accountable. We want to be reliable. Often, if you ever hear from us, this is where you hear from us after you start a project. The RNA-seq is degraded. We wouldn't suggest this, or the DNA is too fragmented, things like that. And this is just giving you a sense of, if you ever get something like this, we usually stop the project immediately and call you and say, we would not advise doing this. But that's what you should rely on, right? And someone with experience to say, oh yeah, we have seen this before. Yes, you're working on FFPE tissue. It's not surprising. You want to do a different type of assay. But this is where our expertise helps. We've seen so many different things, and you would be surprised by some of the things that I've seen. Nothing can surprise us anymore. And so that's your benefit. This is just more specifics. We have, like I told you before, a lot of our work is high throughput sequencing. And these are some of the methods in the main buckets that we have. And again, we have standard analysis, custom. Usually we meet you where you are. If you have a specific question or an analysis you want, or a paper that you saw, that you say, I want to replicate that, we usually can help. But you got to contact us, and depending on the scope of it, we may punt it to Derek or Matt and have them help with that. Lastly, anything in cancer research or any biomedical research is no longer you're going to do RNA-seq and be done. Often you're doing multiple types of omic assays. So a lot of this emergent multi-omic research is where the field is going. It actually is right now, where you will have to do multiple things, multiple omic technologies, whether it's RNA-seq, single cell, spatial, or even just epigenetics. You're going to do ATAC-seq to look at chromatin accessibility. But then you're going to do RNA-seq to say, well, does that make sense? Do I have transcripts that are being made or not, based on what my prediction is? All of these require data integration. And it's an important concept because this is just showing, and this is a few years old now, just from single cell sequencing or single cell analysis. These are all the things you can do now. This is a much smaller list than it currently is right now, and it's growing. And now when you think about bulk RNA, single cell to look at the single cell contribution, and then spatial to try to orient those cells in the spatial context of a tumor, you need to be very, very savvy in informatics and being able to integrate those data to really draw conclusions from that. You want someone to partner with you and help you go through that. And you want someone that has those values that you share and is super experienced in a lot of different aspects of genomics. And that's what Wilmot Genomics is. And that's what we try to build. And that's what we try to start. So with that, that was a real quick snapshot of genomics. I'm happy to take questions and talk about services or other things. I just realized I didn't tell Lindsay I started, so hopefully she recorded. Okay. So with that, thank you. I'm happy to talk, ask questions, or you can get something more to eat on the way out. Thanks. Yeah.
So when you look at the analysis,
Mhm.
I would, we're going to look at analysis, what services are provided, and is it only compatible with like data that you're already collecting before? So like you mentioned a lot, spatial transcriptomics, and epigenomics and stuff like that. What if you have proteomics and metabolomics that you want to compare, things that you might not be maybe as well versed in?
Yeah, great question. That's a partnership with bioinformatics, biostatistics. So metabolomics is a perfect example. We're a genomics lab, we do a lot of genomics data, but we have a lot of skills that are transferable. So Marlies Rossman, PI and DMG, Jalu, who is back there, she's put her hand up, is helping kind of get into proteomics. And we're working also with the biostats folks, Song, in the bioinformatics, shared resource is working with Jalu to also kind of do that. So we're building as the need emerges. Unfortunately, there is no bioinformatics core, right, that does all of this. And so we're leveraging where we've started and the value that we've created, and we're branching out. Metabolomics, proteomics, flow, things like that. But I won't lie, genomics we're very heavy in, and we've done that for 10 years. So right now that's where we're at.
Of course. Thank you.
Sure. Any other questions? Yeah, Jim.
I've seen it grow over the years, which is phenomenal,
Mhm.
and it's nice to have some more bioinformatics support now, but, Les, can you speak a little bit to the integration with Flow, for and the single cell analysis that are doing that?
Sure. So single cell sequencing or the single
Yeah.
cell?
Yeah, single cell, but in terms of the coordination
Yeah.
between getting the cells from the Flow core and then getting the antibodies.
Sure. Yeah, no, that's a good question. So over the years, we've worked with the flow core to identify processes and put things in place to try to minimize issues that happen from coordination or just things like that. The flow now, when you go on and sign up for a cell sort, there's a question that will just go to genomics. And that generally will kick in the background, other specific considerations that we've worked with flow to make sure they actually operate on. It's not perfect, but it works fairly well. Generally, we'll rely on the submitter, whoever the person is that's going to work with us, to let us know when the sort's going to be and things like that, so that we're ready. We've even actually gone over and sat next to the sort operator and just sort of finishes to grab cells. If it really comes down to the comfort level of the person, there are times I'll go over there and set up the gates because I have experience with that too. So we try to be flexible, but there are some processes in place to try to enable and reduce the barriers for people. Yeah. Any other questions? All right, well, I'll stick around if anyone wants to talk about anything else, but thank you.